Switch gears a little bit because I'm a neuroscientist, so I'm really going to talk about the brain and about mechanisms in the brain that are relevant for learning and memory. So in my group, we want to understand how we acquire information, how we store information, how we use information, and how this is being modified by stressful experiences. So in our life, we are regularly exposed to stressful experiences, and that can vary from small daily hassles to really major life-threatening events, and you see a couple of examples here. And we need to cognitively adapt to these stressful situations, and most people can do that pretty well. For example, we remember these events pretty well, which is important for future events, future occasions that we can then better cope with. But at the same time, exposure to stress and trauma, of course, is also an important risk for developing disorders and cognitive decline, as Erik also mentioned. So we want to understand the mechanisms, how we learn, how we remember, how this is being modified by stressful experiences, and also where the risk comes from. And we, in that aspect, really focus on experiences also early in life. So, to do that, from the neurobiological point of view, we study behavior in rodents because we want to understand how information is being perceived, how information is being stored, and how information is being used. In the brain, we focus on those cells that are part of the memory trace. Which cells are activated during learning? Are these cells becoming really part of the memory trace? I'll come back to that later. And how is that being modified by stress and stress hormones? And we focus on the role of synapses, the communication between nerve cells, which is critical for learning and memory. So all in all, this is about adaptation to stressful experiences and plasticity. How does our brain react to stressful situations, and how does it relate to changes in behavior? So, this is basically the take-home message, and these are the points that I will address in my talk. I will discuss how we remember stressful experience so well and show some evidence that we have that this is being mediated by stress hormones that act at the level of synapses. I will also show some evidence that we have that stress and stress hormones regulate the quality of the memory, the generalization of information, and that occurs via the cells that are activated during the learning process. Those are the Engram cell ensembles. And I'll show some preliminary data that we have on the role of immune cells in the brains, microglia cells, which are sensitive to stress and stress hormones, and which seem to play an important role also in stress effects on learning and memory. So we focus in our studies to a large extent on the role of stress hormones, and in particular on the role of corticosteroid hormones, which are being released from the adrenal cortex in response to a stressful experience. And these hormones are very lipophilic. They easily cross the blood-brain barrier, and they bind to receptors in the brain, and there are a number of areas that are very sensitive for these stress hormones, such as the prefrontal cortex, the amygdala, and also the hippocampus. And we focus in our studies to a large extent on the role of the hippocampus. How do these stress hormones regulate processes in the hippocampus in relation to learning and memory? So we can study that in rodents, for example, in very relatively simple behavioral tasks. So this is a simple study in mice where we expose mice to a certain cage. They explore the cage, and they receive a mild negative stimulus in this cage. And we can test whether the animal really remember that a negative experience occurred in this particular context. So by looking at the behavior of the animal 24 hours after training, and we look at freezing behavior. And then after training, if we add corticosteroid hormones to the animals, we find that freezing is enhanced, indicating that the contextual memory is increased. We can also train animals and give an antagonist of one of the receptors of corticosterone, that's the glucocorticoid receptor, and then we find that blocking this receptor and blocking the action of corticosteroid hormones reduces contextual memory, all indicating that stress hormones, corticosteroid hormones, are critical for memory-enhancing effects of stress. So these hormones, they can enhance memory consolidation. Then the question, how does that work? So for that we look in the brain. So we look basically at the communication of nerve cells, Here you see a green cell, here you see a green cell, here you see a blue cell, and we look here at the synapse. And this is a cartoon of the synapse with the presynaptic terminal, with vesicles, with a neurotransmitter, postsynaptic terminal, and we look in particular at the receptors which are present at these synapses. And we look at the role of AMPA receptors, a certain class of glutamate receptors. These receptors are present in vesicles. They can be exocytosed to the membrane of the neuron, and they can, via lateral diffusion, reach the synapse where they contribute to synaptic transmission. So we look at the strength of synapses by measuring the function and the properties of these receptors and how sensitive they are to stress and stress hormones. And this basically summarizes work over the past year. So what we see, for example, is that these receptors are highly mobile, so we can do high-resolution live imaging. This is a dendrite, there are spines, there are synapses, so you can take a look here. Here you see in white basically receptors which go on and go off of the spine and of the synapse. So these receptors are highly mobile. We can measure the properties electrically of these receptors, and we can look at behavior. And to summarize it, we find that stress and stress hormones increase the mobility of these AMPA receptors. They increase the retention of these receptors. They stay longer at the synapse. So really synaptic strength is being increased. We also find that as a consequence of these stress hormones, the connectivity between cells is changed because there are more spines, really structural changes. And these effects are really being modified by the glucocorticoid receptor and via certain second messenger systems, like the mTOR pathway. So stress hormones regulate the strength of synapses, regulate learning and memory via very specific mechanisms by targeting these synapses. Then an important question is, okay, stress increases the strength of the memory, but does it also increase the quality of the memory? And there's evidence also from the human literature that stress can result in memory generalization. And the question is, can we really trust our memories if we're exposed to stress? So we wanted to investigate it also in rodents, what are the underlying mechanisms. And this is a very simple cartoon, what we want to study basically. So if we're exposed to, let's say, two objects which are completely different, like this house and this house, people can easily discriminate between these two objects. But it becomes maybe more difficult if you have something like a house that's more comparable to this one. So you can study discrimination, the capacity to discriminate between similar objects. And the question is that being modified by exposure to stress? And based on the literature, there is some evidence that memory generalization can occur as a consequence of exposure to stress and stress hormones. So we wanted to investigate that and the underlying neurobiological mechanisms. So we can do a similar experiment as what I just explained. In rodents, we can train mice in a context. They receive a mild negative stimulus. We can test them in the same context as where they are trained. And then we can also put them in a completely different context. That's context B. That's a neutral, non-aversive situation, and see how the animals respond behaviorally to this novel context. And we have two groups, control group and a group that is injected with corticosterone. This is a complex picture, but the message basically is that if you look here in this very similar context as where the animals were trained, there's no effect of the stress hormone. But if you place animals in a completely novel, non-aversive context, then the animals that have received an injection with corticosterone, they freeze more. They show an enhanced sensitivity to this context. They should not freeze there. There's no reason to freeze at this context. They freeze more in this neutral, non-aversive context as an index of memory generalization. And then if you expose the animals to the tone, that's what we do here, also there's an increase in memory and freezing behavior. All suggesting that these stress hormones increase memory generalization. And these effects of these stress hormones can be blocked by blocking the action of the glucocorticoid receptor, again indicating that the glucocorticoid receptor plays an important role of these effects. That was shown in this data. I will not explain that in more detail, but the glucocorticoid receptor seems to be important for the effect of corticosterone on memory generalization. Then the question is, how does it work? And for that we look in the brain and we look at which cells are activated while the animals learn. And those are the cells that we call Engram cell ensembles. So here you see part of the hippocampus. This is the dentate gyrus, and you see here beautiful green cells with beautiful dendrites. And these are the cells that were activated as a consequence of learning. And using specific techniques, we can identify these cells, we can follow these cells over time, and really, for example, investigate the special characteristics of these cells. So the question is, do stress hormones regulate the number and the properties of these Engram cell ensembles in the hippocampus? A very simple question. And they do. So we can train animals in a learning and memory task. We can give them corticosterone. And you see that after training, and this is 24 hours after training, we see a couple of cells in the hippocampus. But the animals that have received an injection with the hormone, the stress hormone, which induces fear generalization, there's much more cells are being activated in the dentate gyrus. It's a long-lasting effect. So stress hormones increase activation of the hippocampus. We were actually surprised about it because we would have expected maybe the opposite, but this is what we find. And I think it's also conceptually an interesting finding. So we find that stress hormones increase the number of activated cells in the hippocampus. They not only increase the number of cells that's activated, they also increase the reactivation of these cells. So we can follow these cells over time and we can see whether these cells also reactivated when the animals are exposed to a similar context or a different context than where the animals were trained in. So we can train animals here. We can place the animals in the same context or in a neutral, non-aversive context, and we look at the number of Engram cells, and we can also look at the activation of these cells in these specific contexts. And we find that's changed in animals that have received corticosterone. That's what we see here. So animals are exposed to context A, that's a context that the animals know. We can look at the activation of cells, no effect of the hormone in this case. But if the animals are placed in a neutral context, they have a slightly reduced, lower activation, except when the animals have received an injection with the stress hormone corticosterone. So there's also an increase in the reactivation of these Engram cells. So we find that if you train animals and you expose them to stress hormones, they have an increase in the activation of the dentate gyrus, an increase in the number of activated cells, but also reactivated cells. And then we wondered, can we suppress the activity of these cells in the dentate gyrus, and can we rescue basically fear generalization? That is the question that we addressed. And using molecular techniques, I will not go into that in too much detail, using very specific molecular techniques, we can inhibit selectively the cells that were activated during learning and also during reactivation. And that's what you see here in this case. So, and these animals have fear generalization. They have been injected with corticosterone in the red signals, red circles. And then if we inhibit these cells, those are the blue circles here, we can reduce fear generalization. So we can reduce freezing behavior, suggesting that if we suppress the activity of cells in the dentate gyrus, we can reduce fear generalization and we can make the memory more specific again. So far we have evidence that stress hormones increase memory generalization. They increase the number of activated cells in the hippocampus, all dependent on the glucocorticoid receptor. And if we inhibit these cells, we can restore memory discrimination. The memory becomes more precise again. This allows us to investigate a couple of interesting things. For example, what makes these memory cells so specific? And so you can train animals, as I just explained, and we can identify the cells. So you can do a couple of interesting things. You can look specifically at the properties of these cells electrically. Are they electrically different from neighboring cells, which are not in the memory trace? They are, that's what we know. But we can also look at, for example, at the molecular profile of these cells. What's happening with proteins? What's happening with genes in these cells and neighboring cells? So as a neuroscientist, I think those are really interesting questions. So we have been looking at the molecular profile, not only of these cells, but also of surrounding cells. Very briefly, what we did, we can identify different cell types in the brain. There are different cell types in the brain, not only neurons. There are also microglia cells, astrocytes, macrophages, All cells that also are important for the function of cells, neurons, in the network, and at the end also for learning and memory. So here you see a couple of cells in the brain, microglia cells, astrocytes, oligodendrocytes, all relevant for appropriate brain processing. And what we find is in particular that these microglia cells are very sensitive for stress and for stress hormones. So you can train animals in a fear conditioning task, you can give them corticosteroid, and we see a strong change in the gene expressions, particularly in this group. And we wondered, why is that? Are these microglia cells sensitive for stress? And is it also maybe relevant for how stress modifies learning and memory? So here you see microglia cells in the brain. These are actually beautiful cells, cell body, large extensions. Here you see a reconstruction of a cell, and they engulf neurons. They engulf spines, they engulf synapses, and they regulate the function of synapses. And there's also recent evidence that these are immune cells of the brain, that they regulate learning and memory processes. So we tested whether these cells are relevant for the effects of stress on learning and memory. We trained animals in the sphere conditioning task. We looked at behavior, as just explained, and we used animals which do or do not have a glucocorticoid receptor specifically in these microglia cells. So you see a microglia cell, and with green you see the glucocorticoid receptor, and here you see animals which have microglia cells, but there's no receptor present in these microglia cells. And it regulates learning and memory. That's what you see here. So we looked at freezing behavior. Again, control animals, hardly any freezing behavior. Stress hormones increase freezing behavior. And this memory-enhancing effect of stress and corticosteroid hormones is absent in animals which have no glucocorticoid receptor in the microglia cells, suggesting that also microglia cells are relevant for how stress can regulate learning and memory. So together, to wrap this up, so what I showed you is that the synapses are very sensitive to stress and to stress hormones. Corticosteroid hormones increase synaptic strength by regulating the properties of receptors at these synapses. Stress hormones also regulate memory generalization. They make the memory less specific, less precise. Everything that goes via changing the size of the Engram cell ensembles in the hippocampus. And some preliminary evidence that we have that also immune cells are immune cells in the brain are very sensitive to stress and stress hormones, and they play also an important role in the effects of stress on learning and memory. So that was my last slide, and I'd like to thank a lot of people who have been doing the work in Amsterdam at the SILS, Erik mentioned, and this is the group at the moment, talented student, PhD student, postdocs who did the work. I want to thank you for your attention.